Okay, hello everyone. Good morning in the West Coast and good afternoon in the East Coast of North America, and good night in Europe and Asia. So today we are very pleased to have Dr. Mohamed from Aspect Biosystems today with us. That is going to talk about bioprinted therapeutics, next generation cell therapy. But before going to his talk, like always, I wanna actually review some housekeeping notes. If you have any question, please put it in the Q&A box. Ask a question box on the bottom of your screen. This talk will be recorded, and then you can watch it later, and also, kind of share it on your social media to others. Most simply, yeah. So our next seminar also is next Wednesday at noon Eastern Time with Dr. Emilio Alarcon from University of Ottawa and Ottawa Heart Institute. And if you want to get the most updated information about the seminars, please follow us on Twitter with the handle Translational BME. And like always, we would like to thank our sponsors, Montreal Transmedtech Institute. And for this special talk, we would like to also thank MedTech Talent Accelerators from McGill and Ryerson Universities. By that, I would like to introduce our great speaker today, Dr. Mohamed. A bioengineer, inventor, and entrepreneur. He founded Aspect Biosystems in 2013, currently serves as president and CEO. As a leader and visionary in the field of regenerative medicine and 3D printing, he has been invited to speak on venues ranging from TED Talks to industry, scientific, and executive conferences. He was awarded with the British Columbia's Top 30 Under 30 Award for demonstrating excellence in business, judgment, leadership, and community contribution. Dr. Mohamed served on the board of directors for the Stem Cell Network in Canada, an organization focused on building Canada's stem cell and regenerative medicine research sector. By that, I would like to invite Dr. Mohamed to share his screen and start his talk. Thank you so much.
Great. Thank you so much, Human. And just before I begin, I just want to make sure that you could see my slides.
Yes.
Okay, great. Excellent. Well, thanks very much. Again, my name is Tamer Mohamed, CEO of Aspect Biosystems. Really appreciate the introduction and the invitation to be here today to tell you more about what we're doing at Aspect Biosystems to create next generation cell therapies. So I'll focus most of my talk on key considerations that we think very deeply and constantly about in developing translational technology and science to create impact in regenerative medicine. But before I do that, I was asked to very briefly tell you more about how this all started for me and how I got into this field. So I think it's safe to say that I've been a nerd from the very start of my journey here on Earth. Here's a picture of me in 1992, nerding it out on our very first computer. This was a picture that was part of a campaign run by Science World in Vancouver, aimed at fueling the development and encouragement of even more nerds. So while it may not have been cool at the time, I believe it's never been cooler to be a nerd than it is today. And so nerds are behind some of our greatest discoveries. I don't think I have to convince all of the people that are tuning into this talk of this, but it's just worth mentioning that nerds are behind some of the greatest progress that we're making as humans, from coming up with a COVID-19 vaccine in record time to the many other global challenges that we're addressing through science and technology. Nerds are behind many of these accomplishments. So I really believe our future depends on more and more nerds in science and engineering. So fast forward a few decades after that picture, and my innate engineering tendencies came head to head with the really fascinating world of biology. So I was trained in my undergraduate years as an electrical and computer engineer, but was pulled very quickly into bioengineering during my graduate study research. So to me, the idea that I could trade in the electrical components like resistors and capacitors that I was using to build electrical systems, the ones and zeros that I was using to program computer systems, if I could trade those in with cells and other biomaterials to engineer and create living systems, that would be fascinating, and it really blew my mind. And so transitioning from my undergraduate studies, I embarked on focusing on bioengineering in my graduate studies. Specifically, I was focused on developing advanced 3D printing technology to create living tissue structures. And so here's a picture of Simon Bayer, Aspect Biosystems' current Chief Technology Officer, and myself next to one of our very first early prototypes way back when we were still in the lab at the University of British Columbia. Now I made an initial shift from engineering to bioengineering, and my next shift was into the world of business. And so how that all started was through a fortunate circumstance where the University of British Columbia started an entrepreneurial initiative called Entrepreneurship at UBC. And through this initiative, they wanted to really support aspiring entrepreneurs in science and engineering to take their ideas global and to make a big impact through entrepreneurship. And so through that initiative, we were introduced with a handful of different key lessons that really put my career onto a totally different trajectory from the academic path that I was originally on. And one of the most important lessons that I learned through this transformative experience that I wanted to share is a very simple one, and that is get out of the building. And what I mean by that is the answers to your questions, the answers to many of the problems that you're trying to solve exist outside of the lab. You really have to go talk to people, talk to your customers, talk to your partners, talk to the market directly, listen to key opinion leaders, really understand the problem and make sure you're solving a real big problem. And then going back to the lab and continuously iterating based off of that feedback. And the quicker and more effective you can make these cycles of learning, as I like to call them, the more you increase your chance of success. So a book I recommend to all of the aspiring entrepreneurs on the subject is The Lean Startup by Eric Ries, and it's one that had a big impact on my approach to entrepreneurship. So leaving campus in 2016 from UBC, we've been growing very significantly as a company. We've assembled a team of more than 50 innovators and entrepreneurs, and we're rapidly growing and hiring actively. And we've also been fortunate to be collaborating with a rapidly growing number of leading research and commercial organizations around the world. These are some of the biggest names in our industry, from GlaxoSmithKleinMerk to Merck, Johnson & Johnson, and other groups that we've been very fortunate to work with on different applications of our technology. So just as a briefer on Aspect, our company is focused on combining our microfluidic 3D bioprinting technology with advanced biomaterials and cell biology to create the next generation of cell therapy in the form of implantable bioprinted therapeutics. So what does that mean? It means that we're applying our technology platform internally to create a pipeline of implantable bioprinted therapeutics that we are driving to the clinic. But we also partner strategically with leading innovators, both in industry and academia, to tackle different applications beyond our internal pipeline and really to address some of the toughest problems in regenerative medicine. So you could have the greatest technology in the world, but if the timing doesn't make sense, you're going to have some difficulties. And the good news is the time is right for this type of technology. And so we're seeing a major paradigm shift in how we think of drugs. Small molecule drugs were once seen as mainstream, but the market for biopharmaceuticals, where the drug itself is made of biological substances like antibodies, is rapidly expanding. And now I believe we are entering into the exciting era of regenerative medicine, where we're seriously starting to look at cells and tissues and even one day organs as the next generation of therapeutics. Similarly, we're seeing a major paradigm shift in the medical device world. Medical devices are becoming a lot more sophisticated and even personalized, and advanced manufacturing like 3D printing is a big driver behind the shift. And so I ultimately see these major paradigm shifts both in the drug world and the medical device world converging to enable the next generation of therapeutics in the form of advanced manufactured tissues. And I think 3D bioprinting is a big part of that. So at Aspect, we're focused on marrying our microfluidic bioprinting technology and manufacturing platform with advanced cell biology and fit-for-purpose biomaterials to create bioprinted therapeutics. And so what are these therapeutics? They are implantable cell-based therapeutics that we believe will unlock the next generation of sophisticated cell therapies. And so at the very core of our technology is our microfluidic bioprinting platform. So we pioneered the marrying of microfluidics with 3D printing technology, and that allows us to have a very deep level of microscale control over the biology. So with our platform, we can integrate, process, and print multiple different biomaterials loaded with therapeutically relevant cells in real time to create living microscale fibers. So think of these fibers as the building blocks of our implantable bioprinted cell therapies. So these fibers are highly customizable. For example, we could rapidly sequence between different core and shell materials to provide multifunctionality. We could print these fibers with a hollow core to enable direct perfusion. We could also print fibers with overall diameters ranging from as small as 10 microns to over a millimeter. And so we have a lot of flexibility to design these different building blocks for our tissues. So these example capabilities provide a powerful toolkit for rationally designing structures that promote cell viability, but more importantly, function of these tissues. And so these dispensed and cell-loaded fibers are then directly patterned into 3D macroscale therapeutic devices. So using our advanced software design tools, we are able to rationally design both the structure and composition of these bioprinted therapeutics to meet different functional requirements inside of the body. And so at Aspect, we are focused on creating a global network of collaborators to tackle different applications. And we work very closely with these partners to advance the most promising opportunities into commercial and clinical outcomes. And so here's an example of some of the global innovators we've been fortunate to partner with. By combining our platform technology with domain experts, we really believe we can more quickly unlock the broad applicability and potential of our bioprinting technology. So I'll quickly highlight a few examples of the impactful applications our partners are focused on, starting with Maastricht University, where Professor Moroni and his team are applying our microfluidic technology to create perfusible renal tubules. Initially, this work will be used to better understand kidney disease and be used as a disease model for drug discovery and development. But in the future, we believe this technology could potentially be applied to creating implantable therapeutics for different diseases that are relevant to the kidney space. Another example is at the National Research Council here in Canada, where our partners are using our technology platform and working side by side with our team to attempt to break through the blood-brain barrier, one of the biggest hurdles in getting therapeutics to the brain to treat neurodegenerative diseases. So the goal is to create a 3D bioprinted model of the blood-brain barrier, and early results give us a lot of reasons to be excited about this approach. Another example partnership is with GlaxoSmithKleinMerk and also McGill, where we are taking patient-matched tumor, stromal, and immune cells and bioprinting these different cells using our microfluidic technology in controlled microenvironments to discover new anti-cancer therapeutics using this high-throughput disease modeling platform. And so these are just a few examples, but there are many, many more amazing applications being explored through our global partnerships. And I would invite you to reach out to us if you have an interesting application that you'd like to explore through a collaboration. We'd love to hear more, and we'd love to see how we could support your research and partner to unlock new possibilities in the space. So the rest of my presentation will focus on how Aspect is focused on applying our technology to create living bioprinted therapeutics. So in addition to our partnering at Aspect, we're developing a pipeline of bioprinted therapeutics. So I thought it would be useful to share with you some of the key areas of consideration that we think very deeply about to ensure translation into the clinic. Starting off with designing your target product profile. You could have a platform, but if that platform doesn't address a particular unmet medical need with a specific definition and description of a product, that platform really doesn't mean anything. The platform is only a means to an end, and so the end is the product. So really defining your target product profile upfront is absolutely critical. And then there needs to be a focused attention on understanding the raw materials that are going to make up your product. And in the context of bioprinted therapeutics, those raw materials include the cells and also the biomaterials. Developing a controlled process for manufacturing your product, understanding the regulatory pathway, and finally, developing your clinical adoption strategy. So I'll focus primarily on the first three areas using a case study of a very specific bioprinted therapeutic that we're developing at Aspect. So starting off with designing your cell-based or bioprinted therapeutic, I want to touch very briefly on this subject. So one traditional design approach in tissue engineering is a top-down approach where the goal is to create a structural replica of an organ. So our process at Aspect is a little bit different than that in designing bioprinted therapeutics, and that it really starts with identifying the unmet medical need and deeply understanding the underlying cause of the disease or the biological malfunction. Then we focus on rationally designing a biofunctional implant from the bottom up rather than the top down to address that biological malfunction. So in this way, our design philosophy is really focused on function over form. So one example of a bioprinted therapeutic platform currently in development at Aspect targets indications where allogeneic cell encapsulation is critical. So using an immune protective outer shell shown in blue, we were working to enable long-term allogeneic cell therapies without the need for patient immune suppression. So we use advanced biomaterials that are perm-selective, which enables a sense and release function by the encapsulated therapeutic cells. From a safety perspective, we are developing this platform as a retrievable implant. We believe that being able to retrieve a cellular device, especially if it contains a stem cell source of therapeutic cells, would reduce the risk of cells escaping from the device and potentially forming teratomas. From a customization perspective, one of the powers behind our bioprinting approach to cell therapy is that these implantable devices are printed by design using our platform. So things like sizing and patterning of the macro device can be altered to different therapeutic cell doses or to encapsulate cells at lower or higher densities to optimize functionality depending on metabolic demands of the cells. Device design is also driven by surgical procedure and site of implantation. And by using different cell types, our goal is to use this bioprinted therapeutic platform to generate customized bioprinted therapeutics that could potentially address multiple different indications. And so we're currently applying this bioprinted therapeutic platform to several indications, including type 1 diabetes, as well as acquired and genetic liver disorders, as well as a host of other applications and indications that are at the discovery stage. But for the purpose of this presentation and highlighting the several things that we think about when developing a bioprinted therapeutic, I'll focus entirely on our pancreatic tissue program in the interest of time. So the Edmonton protocol is something I'm sure many of us are aware of, and it has shown great promise and validation of the potential for cell therapy to cure patients with type 1 diabetes. But there are limitations with the Edmonton protocol, and that includes a limited availability of donor islets and the need for long-term patient immune suppression with associated risk factors. So our bioprinted therapeutic platform has the potential to overcome these hurdles by encapsulating a scalable stem cell source of beta cells with immune protective, perm-selective biomaterials patterned into an implantable and retrievable 3D device. So the engine of your cell-based therapeutic is really the raw material, but very importantly, a key determinant of the success of your product. And so these cells are an essential component of these bioprinted therapeutics. And so there are key considerations to make when selecting what are the most appropriate cells for your product. Primary cells versus stem cells, autologous versus allogeneic. Each of these decisions have very important implications. So for example, primary cells could reduce biological risk as you are certain what those are. They are terminally differentiated cells. You know what they are, and you know what you're dealing with. but you also have limited scaling potential, and these cells are also very typically expensive to incorporate into a commercial product. A stem cell strategy, on the other hand, could scale much, much better, but differentiation protocols require deep amounts of validation and also intellectual property protection. Autologous solutions offer the benefit of personalization and evading the immune system, but don't have as strong of a scaling potential offered by an allogeneic strategy. So in all cases, ensuring that the cells you're using have a path to the clinic is absolutely critical and needs to be considered upfront in the development process. And so for our type 1 diabetes program, one of the cellular sources we are using include primary human beta cells. On the top right, we show that tissues printed with these cells are functional using a glucose-stimulated insulin secretion assay. Now, freshly isolated islets vary very heavily in size, making them difficult to print consistently. And so larger islets are also prone to developing necrotic cores. And so on the bottom, we show that function can be improved by using what are known as aggregates or islet aggregates or pseudo-islets. And so these are uniform cell clusters formed by enzymatically dispersing primary islets and then reaggregating them. So looking at the viability of the islets and reaggregated islets at 7 days post-printing, as shown by fluorescent live dead assay, we see almost 100% viability. The pseudo-islets are a more homogeneous distribution within the printed fiber, and function as measured by glucose-stimulated insulin secretion at 7 days is improved in the reaggregated islets compared to the bioprinted primary islets. Now, human islets have been demonstrated to be a clinically relevant cell therapy for treating type 1 diabetes. The Edmonton protocol demonstrates the efficacy of primary islet cell therapy in severe uncontrolled diabetics, but limited applicability due to the requirement for long-term immune suppression with unencapsulated allogeneic cells. And so we spent significant time optimizing the bioprinting process, the viability and functionality of primary islets in vitro, and the data that I'm showing here today is from in vivo studies that is built off of this early work. So 3,000 islet equivalents were implanted into the interperitoneal space of healthy immune deficient mice for 31 days. Bioprinted tissues retrieved at 31 days demonstrate high viability, confirmed via live dead stain, and insulin content confirmed via dithazone staining. We see good functionality as measured by glucose-stimulated insulin secretion before as well as after 31 days of implantation. At 31 days before tissues were explanted, blood levels of human C-peptide as well as glucose were measured at regular intervals for 60 minutes after an oral bolus of glucose. And we see that C-peptide increases rapidly in animals with bioprinted human islets with a corresponding rise, then rapid drop in blood glucose. So we think a beta cell therapeutic that immune protects functional cells and can be used without systemic immune suppression could expand primary islet cell therapy to a broader number of patients. However, there are an estimated 1.5 million type 1 diabetics in the US alone. So we believe, and we're very mindful that primary islet cell therapy alone is not going to scale to this many patients. And so that's where we also are addressing this using stem cells. So we're working with one of the most well-known groups led by Professor Tim Kiefer at the University of British Columbia in Vancouver, Canada. So Professor Kiefer was one of the first to develop a multi-stage protocol to differentiate embryonic stem cells into pancreatic islet cells. And so we printed with stem cell-derived cells at stage 6 and almost at the final stage of differentiation, but not yet capable of releasing insulin. And after 7 days in culture post-printing, GSIS shows bioprinted stem cell islets have become functional, suggesting that the bioprinted 3D microenvironment is capable of supporting the final stages of beta cell differentiation. Stem cell islets implanted into immune deficient mice are viable and arise in circulating human C-peptide levels in animals with stem cell islet tissues for over 50 days shows the stem cell islets are maturing into functional insulin-producing cells in vivo, and you can see that here on the graphs. So the raw materials include not just the cells, but the biomaterials. So the biomaterials will have a direct influence on the properties and the translation of your therapy. And there are several considerations to study very carefully when selecting your biomaterial. compatibility with your cells, consistency, mechanical properties, biofunctionality. Are you going to use something synthetic versus naturally derived? What is the associated supply chain considerations? How scalable is that source? What's the cost? What's the history of use in other approved clinical products? And so your biomaterial choice heavily depends on your application. And we really believe that there isn't just one single biomaterial or bioink that addresses every application. So in this particular application, we're using an alginate-based biomaterial in our type 1 diabetes program. So this is a naturally derived material that has very well understood biocompatibility through decades of in vivo and clinical validation. From a supply perspective, the material is abundantly available, including in GMP grade. Cross-linking modalities are non-cytotoxic and enabled with Aspect's microfluidic technology. From a function perspective, the hydrogel can be tailored to be perm-selective. This allows diffusion of oxygen and nutrients into the 3D tissue and a fusion of therapeutic soluble factors out. The material also has strong potential for immune protection. In vivo studies in immune-competent rodents confirm the alginate shell provides immune protection of therapeutic human cells from the host immune system. In terms of foreign body response, multiple strategies have been shown to mitigate this, including architectural and alginate modifications. And from a mechanical perspective, tests performed before and after in vivo implantation to examine tissue stability confirm suitability for implantation. And so here we demonstrate data from studies that involve implanting our alginate-based eyelid tissues in disease models that have been triggered to become diabetic animals by exposure to injected streptozotocin at 5 days. Blood glucose rises very quickly as the mouse beta cells die due to STZ injection. And if we look at our bioprinted tissues containing 3,000 IEQs, they were implanted into the IP space at time 0, and we see very rapid return to normal glycemia by 10 days. Return to normal glycemia is also associated with a rise in blood levels of human C-peptide, and we see that normal glycemia and C-peptide of over 1 nanogram per ml is maintained for at least 60 days. So to investigate in vivo function of our bioprinted islets in diabetic mice, we performed high-resolution blood glucose measurements post-glucose infusion at 32 days post-implantation, and we demonstrate rapid return to normal blood glucose levels in animals with bioprinted islet tissues, suggesting appropriate kinetics of insulin release from the implanted tissues. And so through our studies, we confirmed that our bioprinted islets at 68 days post-implantation demonstrate high viability and improved function. Tissues retrieved from mice at 68 days post-implantation show close to 100% cell viability and intense dysosome staining, suggesting very high insulin content. And GSIS measurements of explanted tissues demonstrate several key takeaways. The first is an 8-fold increase in their stimulation index compared to pre-implantation, reduced variation in glucose-stimulated insulin release in explanted tissues versus tissues pre-implantation, and very low basal levels of insulin release and return to similar low baseline levels when glucose is reduced, which is a very good sign of islet health, as stressed islets have higher basal levels of insulin release. So I know that was a lot of information very quickly, and so to take us back to a 90,000-foot view and looking at the big picture, really what we're focused on doing is marrying our microfluidic bioprinting technology and manufacturing platform with therapeutic cells and fit-for-purpose biomaterials. We're able to directly manufacture these implantable cell-based bioprinted therapeutics. These bioprinted therapeutics are rationally designed with not just functional biology, but also importantly, the necessary architectural and mechanical integrity for handling and for implantation. And so here we show how our bioprinted therapeutics are easily handled and subsequently implanted into the omentum of a pig for evaluation as we enter into later stages of preclinical development as we drive our bioprinted therapeutics to the clinic. So in developing a complex cell-based therapeutic, I really believe that your process is your product and your product is your process. And so once you pass the initial stages of discovery and you think you're onto something, once you have proof of concept, for your target product profile, you really need to understand all the variability in your system. The cells, how you're validating these, the materials, understanding all the components, the assays used to confirm quality, and the processes used to manufacture your product. To be able to close, control, and scale that process is absolutely critical to the success. And so these are aspects we think very deeply about, especially as we enter into this next stage of development. Now, other very important aspects to consider early on include regulatory, manufacturing, and clinical adoption. Deciding what regulatory pathway your product will take is absolutely critical, and doing that upfront is even more important to ensure translation. So speaking to regulatory agencies through early interact meetings will help ensure that your product will be accepted for clinical evaluation. So several agencies, including the US FDA and the Japanese PMDA, have introduced accelerated pathways for regenerative medicine. So it's really important to consider these advantages to your development. So from a manufacturing perspective, there are many decisions that need to be considered. For example, will your product be manufactured at point of care? That could potentially be possible for traditional cell therapies where you're injecting cells. But if you're doing something much more complex, like what we're doing here at Aspect, where we're trying to integrate materials with cells using a new manufacturing process, something like centralized manufacturing becomes a lot more feasible because you have control over that process. And that model also scales a lot more than point of care. So with point of care, you can closely integrate within a healthcare workflow, but it comes with a loss of control on quality as you're relying on others to manufacture. But with centralized manufacturing, you can have more control over quality, but also need to develop appropriate storage, shipping, and logistical processes. So you really need to weigh the pros and cons and decide what makes sense for your particular target product profile. And very lastly, understanding how clinicians will ultimately use your product. I think that input is absolutely critical. Talking to clinicians upfront to see the little details. How are they going to handle these tissues? How are they going to surgically implant them? How are they ultimately going to adopt them? And very importantly, at the end of the day, who will pay for your product from a reimbursement perspective, and who will ultimately sell and market your product? So defining your go-to-market strategy, how far are you going to go before you bring in a partner? All of these things are very important to consider upfront as you move away from just research, but to translational research. So last, but certainly not least, I'd like to acknowledge our amazing team at Aspect. We have grown the team from 4 co-founders, myself and 3 others, that were the only part of the team 4 years ago. And now we have over 50 people, and we're adding about a couple people, even a handful of people every month over the next period. And really our people are tackling some of the biggest challenges in the space and are ultimately aligned around the common vision of using our deep tech platform to create cutting edge bioprinted cell therapies that can make a difference and make an impact on patient care. So I'd like to point out that we are actively hiring, particularly around R&D. We have multiple positions on immunology, biomaterials, cell biology. And if there isn't a position up there, but you're interested in what we're doing, we'd love to hear from you. And we'd love to see how you could help join our mission here to make a tremendous amount of impact. So with that, I will stop there and I'll pass it back to Mohsen and Human for questions.
Thank you very much, Tamer, for your very nice and inspiring presentation, as always. I know Tamer for a long time, and I know how hard working he is. He's one of the PhD students that any supervisor, any PI would like to have. And then we're lucky that he is our neighbor in Vancouver, and then we get to see him once in a while when he visits Victoria. So thanks, Tamer, again. So we'll start with the questions. I encourage the participants to ask their questions in the ask a question box. Tamer will be with us for the next 25 minutes, and then he has other commitments that he has to leave. So we'll start with the first question. It's the first question is from Imran Ahmed, and then it thanks you for your great talk. So they say that I am a MEMS engineer working in the field of fabrication of microfluidics microvalves. So please provide your guidance to have a career in the industry.
Yeah. Thanks for the question, Imran. I think the field of microfluidics and MEMS has, I would say, really skyrocketed the past several decades. And really, I think what's fueling that is people are starting to identify real world applications to these, whether that's using sort of microfluidics for antibody discovery, like Epsilera is doing in town here in Vancouver, or Aspect using microfluidics for bioprinting and using that bioprinting to develop specific applications, or another company in Canada here, Precision Nanosystems, was using microfluidics for encapsulation or delivery of mRNA drugs and nanomedicines in general. I think that the key to all of those initiatives succeeding is identifying what the problem is, and identifying how MEMS could help there. And so I would really encourage you to take your expertise and try to apply it to a real problem, and that could be done in several of these companies that I mentioned, and there are others. So I would really encourage you to embark on seeing what companies are using this technology that could leverage your expertise and applying it in unique ways. I'm sure they'll make great use of your expertise. And I'd also invite you to apply for positions at Aspect out of my own self-interest.
Well, thank you, Tamer. And I wanted to reiterate what Tamer said right in his first slide. So try to be as nerd as possible, because nerds are making the future of science and engineering. And by the way, that was a very cute photo of you, Tamer, in the beginning.
Yeah. I debated with my marketing team for including that, because I don't like to include my baby pictures, but I made my marketing team and probably my mother happy. So.
Well, yeah. So let me just ask the next question, which is from Mariam. So thank you very much for your very nice talk. So the question that Mariam has is about the type of bioprinting technology that you're using. So is it an extrusion-based type, and or maybe how do you mostly about the technology itself, how you manipulate the flow in your microfluidic chips? Maybe a little bit explanation about the technology, if that's possible.
Yeah, so really good technical question. The short answer is we use both modes, both syringe pump or air pressure, and each come with its own advantages. And I'm sure Mariam knows of these, hence asking the question. I think what we have seen that with sort of syringe pump systems, we could get a lot more, I would say, control. But what we're able to do is we're introducing ML-based control systems that are able to really generate real-time readings of our flow parameters, and then developing control systems that are adapting this. And so that's those are smart systems that we're developing that allow us to, in real time, figure out what our parameters are, and then very quickly initiate course corrections to anything that needs to be addressed. So that's a big area of development at Aspect, and key features that our partners take advantage of to ensure control. And this is something that's very important, not just for technology for research, but for us at Aspect, we're using this technology to develop clinical applications. And so making sure that we have very fine control. And so we use a lot of machine vision and machine learning to ensure, for example, we have the right concentricity of our flow regimes. We have fiber diameter measured in real time and adapted if necessary. And so we use those two modalities, and then we apply a lot of sophisticated ML-based and machine vision-based control systems to control a lot of our parameters.
It's great. So the next question is basically there are like two participants asked pretty much the same questions, Magdalena and Red Team. So they thank you both for your very nice presentation. What they want to know is more about your blood-brain barrier model that you briefly discussed.
Yeah, absolutely. So the strategy there is really developing sort of a multi-core shell structure with endothelial cells and neural cells and constructing that and then flowing essentially therapeutic agents through that direct perfusion. And so the team there is making a lot of progress, and they have some amazing data using these endothelial cells and combining that with neural cells. And so that program started about a year ago, and I think they're approaching a stage where they have some data that is publishable. And so look out for that publication in the very near term. I won't steal their thunder, and so I won't reveal too much other than yeah, they're using core shell technology to pattern multiple different types and using perfusion to model that.
Wonderful. We're looking forward to seeing those publications. So the next question is from Jeremy Rota from McMaster. Hi, Jeremy. So great talk, Tamer. Could you expand on the personalized medicine approach / vision? Are you thinking using iPSC type cells and reimplanting them into very individual they were isolated from?
Great, and thanks for the question, Jeremy Hirota. I should mention Jeremy. Is it Jeremy or who is it? His brother? Which Hirota am I talking to?
It was Jay. I mean, I think.
Oh, okay, great. Okay, well, he's also a partner at McMaster University who has our technology, and so great to connect with you again, Jay. Yeah, so I mean, this is a constant philosophical debate that we have internally and externally in terms of personalization. I mean, personalization sounds very good theoretically. It's very hard from a translational perspective. And so a lot of venture capitalists in today's world won't touch a personalized therapy with a 10-foot pole just because it's very hard to scale those types. And so we're really looking at sort of allogeneic approaches now that could be sort of off the shelf type therapies. But let me read the question again just to make sure I'm actually addressing the question. I'm trying to scroll. Could you say that again, the question again?
Answered question, Mohsen box. Answered one. I just
yes, I found it. So it's a so are you thinking about using iPSC type cells and reimplanting them into the very individual they were isolated from?
Right. Yeah, so I would say that would be amazing. Right now, we're using iPSCs and ESC-derived cells. They aren't obviously patient-specific. I think that there is a potential there, but I think it poses a lot of challenges from an economies of scale perspective.
I think we lost Tamer.
Yeah, is Tamer caught? Okay, yeah. I hope he gets back. Because we have wonderful questions from audience. I see all
sort of allogeneic cell encapsulation through sort of off-the-shelf therapies. Oh, did you guys lose me?
Yeah, we lost you. Can you repeat your answer?
Oh, right. Okay. Yeah. So what I was saying is, right now we are using iPSCs and ESC-derived cells. They're obviously not patient-specific. I think from a technology perspective, that's certainly possible. I think the challenges are more so with the process and actually scaling that. So if you can imagine, like with that type of a workflow, you would have to, for example, draw blood from a patient in the hospital, or take a sample of cells from their skin, take those stem cells, derive them into, for example, beta cells offsite, and then integrate them into the tissues, and then do quality control, ship them back to the hospital, and make sure you keep the cells alive during that process. That could take weeks if you're successful, and then the economies of scale become very difficult. And so I think potentially one day, but I think the more, I would say, feasible, scalable approach would be off-the-shelf allogeneic sort of approaches.
Wonderful.
And
thank you very much.
Unless I would say it's something simpler if you're just injecting cells. So
Yeah.
and that's why you're seeing a lot of this in the oncology space. If they're taking cells and doing something with it on site in the hospital, but if you're taking cells, combining that in a matrix, manufacturing that, and now you have a personalized product, it becomes a lot tougher.
Agreed. But these are challenges that I'm sure great
Nerds will solve.
scientists and nerds will solve them. We need more nerds. So Chris Moraes from McGill is asking another question. So thank you for this very cool technology. So I apologize for missing this part, but for the project you described, why would you want to 3D bioprint it compared to just mixing your cells with hydrogel prepolymers in a dish, or droplet encapsulation and packing, or other simpler methods? So the rationale for using 3D bioprinting for some of the technologies that you're using.
Yeah, so really what the bioprinting allows us to do is to incorporate those materials that you mentioned, but incorporate them into a design that we think is going to work. And I think we really look at the bioprinting as a design, but also manufacturing technology. And so in a direct step, you could create these structures that have certain features like optimized diffusion characteristics, optimized architectural arrangements for a particular surgical site. You could incorporate cells in an environment that keeps the cells alive. And so really what the bioprinting is, it's a manufacturing technology that allows us to not just manufacture, but also use the same system to design and then directly manufacture with these technologies. And so, but you're absolutely right. There are other approaches, and these other approaches are approaches that we think very deeply about, and we try to address problems that these other approaches had with in terms of this particular application that I showcased. But we think that the bioprinting technology offers you a ton of flexibility, a ton of degrees of freedom to come up with your most optimal design for the biofunctionality and for the architectural requirements of that particular implantable device.
Great. So the next question is from Karina Martinez. Thank you for your great talk, Tamer. I was wondering if you could briefly talk about the timeline from discovery to manufacturing and production. And lastly, how does Aspect Biosystems collaborate with universities? Do PIs/students approach to your company to start a collaboration, or do you use a third party such as Mitax? Thank you.
Yeah, so for the first part of that question, in terms of the process, you're absolutely right. It starts with basic research, discovery, and then moving into lead optimization, identification of a candidate, and then once you have a candidate, moving through IND-enabling studies, and then ultimately submitting an IND, and then moving into clinical evaluation. Now that time could take a lot of time, and so I think it's really important to just make sure that you have appropriate gates at every stage. And so Aspect has multiple programs now in the preclinical process, but that started with basic research, collaborating with researchers. That's why we try to work with researchers at a very early stage where we could de-risk a lot of the biology, and really at Aspect internally, we're focused on addressing areas where the biology has been de-risked, but we're applying our bioprinting technology to well understood biology. And so these are things that you have to consider in a commercial sort of setting, because you want to ultimately succeed. And I think what the industry needs most now is success in terms of these therapies getting to the clinic. And I think that will really fuel a lot of other people to start. And that's something at Aspect we want to see. We want to see the creation of not just a successful company in Aspect, but a success of an entire industry here. And so the timelines vary from months to years, but I think if you can find ways to collaborate early on and you pick the right indications, you could significantly accelerate these timelines. And then the second part of your question is, how do we work with academics, I guess? So we're very partnership focused, and so we work very closely with academics around the world, and those partnerships emerge from people reaching out to us, but also us identifying opportunities where we need to speak to a particular researcher because we think a partnership makes sense. So I think both channels of communication are important. So I would encourage you to reach out to our team, and we have a team that is dedicated to research partnerships. And then in terms of your last point, I think it was something around MyTax.
MyTax. So you pretty much covered that question. So what are the opportunities in terms of funding? So how do you? Are there any funding mechanisms that you typically use to initiate these collaborations or?
Yeah, so a variety of different grants, and we also do sponsored research collaborations as well. And so I think from our perspective, let's start with discussing what the opportunity is, and then if it's a great idea, it'll find money.
So that's great. So the next question is from Ilan Ismail. So great presentation, Tamer. I see that you are interested in using machine learning for optimizing your manufacturing process. What are the types of projects that this work would entail? And what is your biggest challenge so far with manufacturing?
Yeah, so I think we see an opportunity where this type of approach could have a big value in controlling your process, and we could all, I think, agree that there are multiple variables here in creating bioprinted therapeutics, and with each of those variables, there's a high chance of a lot of variability. And so I think machine learning is one approach, and so that's an approach that we're pushing heavily, and we're actually recruiting people on that team now. And so I invite you to if you're interested in this, to definitely consider that opportunity, and we could discuss it in more details sort of offline.
Okay, great. So I have a couple of questions from undergraduate students. Some of them are from University of Victoria that I see here, biomedical engineering students, and then another student, Marie. So they thank you for your presentation. So they mostly the undergraduate students would like to see how they can be involved in your research activities. Are there any opportunities for them? And then how can they approach you?
Yeah, so we usually have a summer class of undergraduate co-ops that attend. I think every summer at least a handful. We may be close to completing our class for this summer, but we're always open to bringing on additional people. There's not a specific hard line in terms of the number of people. And so in terms of how to reach out to our email that's on our website, and it'll definitely make its way to our talent team that is actively looking for not just full-time permanent positions, but also setting up our co-op teams for the summer and for other times of the year.
Okay, that's great. So those undergraduate students who are on this talk, so feel free to email Aspect Biosystems team. Their email address is on their website. So send many emails to them. That's what just Tamer said. So the next question is from Amir. So thanks for your presentation. Is there any example in which you could introduce as a so if you can use microfluidics technologies that are already in the market? I'm not quite sure if I get this question. So maybe Amir, can you clarify more on your questions while I'm asking another question? So let me just ask another question from Yasaman. Thank you very much, Tamer, for your nice presentation. I recently graduated with my PhD in biomaterials and have experience with bioprinting technology. So they want to see how they can, so people can get positions in your company. People are very interested in getting a position in your company. The technology is very interesting. People are very excited, but I'm sure that you receive many applicants. What is your advice for the applicants who apply to a position in your company?
Yeah. So there's really no hard line on what the experience is that you need, but what we do look for is just a demonstration of excellence in whatever you've done, number one, and a command of knowledge in a particular area that we're looking to address, and just a clarity in providing solutions to the problems that we're addressing. And so, like you said, Mohsen, we're humbled and really grateful that we receive a lot of applications to every single position, and these are the hard decisions that sometimes you have to make. Is there's one position and there's one person that's probably going to fill that, and so what I would say to anybody who's applied to a position is we try as best as possible to provide feedback to people who made it to the very end stages, but ultimately didn't get it. Obviously, we can't provide feedback to everybody, but we try to provide feedback to people who made it to the end stages. And if we haven't done that, I apologize. Reach out to me, and if you did, we'll reach to the end of the process. We would be more than happy to provide feedback. And it may just be that things just didn't work out that one time, but I encourage you to continuously apply. And we're always looking for great talent, because at the end of the day, a company is nothing more than just people, good people, smart people moving in the same direction. So we need people to be interested in what we're doing.
Keep applying. So thank you, Tamer. I mean, this is for any job position that you apply, and keep applying, keep improving your CV, and then you will get there. And I can assure you that I talked to, so Tamer is very good in replying to emails, and then he will, with his busy schedule, he will provide you with feedback. But these positions are very competitive, as Tamer mentioned. So the next question is from Didarul, and I think we have only 4 minutes left. So I'll go with this is going to be the last question, and I will ask my questions later from you, Tamer. I didn't have a chance to ask any questions from you today.
Anytime. Well,
so great
Me
presentation.
too.
Who man too. So great presentation, Tamer. Really enjoyed the talk. Is Aspect interested in tissue manufacturing for clinical implantation? What do you see as the biggest challenge for clinical translation of? I assume that like for these tissue, for these bioengineered tissues. So it's a long question. So maybe you can answer the first 2 questions, and then if you have time, then I'll ask you the rest of the question.
Yeah. So many of the things that I was trying to highlight in the talk, in terms of the process development and your MSAT, your manufacturing sciences and advanced technologies, just showing proof of concept is not enough. And you could show proof of concept, but translating into the clinic is an entirely other beast altogether. And so considering all of those things that I mentioned, what cells are you using? How are you going to qualify these cells? The materials, how are you going to qualify these materials? What materials are you using? What animals are you going to use? What pathway are you going to go down? So these are things that we think of deeply. I would say these are things we think of even more now at our stage of development than the early sort of research types of questions. You need the early research discovery type stuff, but as you start to get closer to the clinic, you need to figure all of these things out and submitting your package to the FDA and whatnot. So these are, I don't know if there's just one challenge, unfortunately. There's many challenges. I guess you could call that one big challenge, but yeah, there's a lot going on, and I think what's key to that is just hiring the right people. And so we have people at Aspect that have taken products into the clinic in the regenerative medicine space. We will be bringing on an executive and several other people under that executive in terms of process development and MSAT. So also look out for that job description as well. And so, yeah, the only way to get through challenges is to bring people who give you a good shot at tackling those challenges. And then the second point, I think I touched on this, centralized versus point of care. So maybe I'll skip that now. I hope I addressed that through a previous answer, but if not, feel free to reach out and I'm happy to share more.
Wonderful. Great. So I don't know if you have more time for another question, Tamer, but if not, then we can conclude this session today.
Yeah, I would be happy to take the last one, and then I'll have to get going here.
Okay, that's great. So so the question is about is there any product which produced by microfluidic systems? So the question is that how big of a role the microfluidic technology plays in your microfluidics plays in your technology?
Yeah, it's a big part, and so we are using it to um, flexibly design different tissues, and so we think it's a big part, but it's not the only part of our technology stack. Our technology stack includes the cells, the materials, the manufacturing platform, and so it's really the heart of our bioprinting technology. But we also have a deep technology stack to address other considerations for regenerative medicine products. I hope I addressed your question, Amir.
Wonderful. Well, thank you, Tamer. So thank you very much for your time. Just before we leave, I'd like to again announce that we will have Dr. Emilio Larcon from University of Ottawa Heart Institute next week. It's going to be a very interesting talk. Looking forward to Dr. Larcon's talk. And then I also would like to thank all the participants for attending this meeting today and asking many good questions from our wonderful speaker, Tamer. And sorry, I didn't have me and Human and I did not have a chance to ask you questions, but it's it was very fortunate for us to have you on in these e-seminar series. With that, I would like to end this seminar today and see you all next week.
Thanks,
Thank you
Masa. Thanks,
so
Oman. Bye-bye.
Bye-bye.